Okay, we can get going. And hopefully we won't have another day of adventures with the Wi-Fi. We'll see how it goes. So where we're headed today is we're going to talk about the components of blood. And we're going to see that when we spin blood down in a centrifuge, we end up with several regions. We'll end up with a region called the plasma. That's going to be about 55% of the volume. About 45% of the volume will be the hematocrit, which is erythrocytes or red blood cells. And then in between those two layers, we get a very thin layer called the buffy coat. The buffy coat contains all the cells of the immune system, and we're going to spend some time on those as we get toward the end of the week. But the buffy coat is going to contain lymphocytes and macrophages and granulocytes, a whole series of cells that are involved with the immune system. That will also contain the platelets. And then we're going to describe something that's fairly unique to our understanding of blood. And we're going to divide the cells of blood into those things which we call true cells, formed elements, and those that are not. So all of the cells in blood are in fact formed elements. So if they're true cells, like cells of the immune system, like macrophages or lymphocytes, those are proper cells. They have a nucleus, they have an endoplasmic reticulum, they have ribosomes, they do all of the sort of general cell machinery stuff that we've talked about in here. But there are other formed elements in the blood, and we use that euphemistic term formed elements so that we don't refer to them as proper cells, because both the platelets and the erythrocytes are not true cells. The platelets, which are really important in blood clotting, are fragments of a larger cell called megakaryocytes. And they contain no nucleus, contain no ribosomes, they can't do protein synthesis. They're basically plasma membranes surrounding a series of vesicles rich in serotonin and a bunch of other clotting factors. They're not really cells. The other major character that we see in the blood, which is about 45% of the volume, are the erythrocytes. Sometimes you'll see those referred to as red blood cells. But the red blood cell term is a bit of a misnomer. It's not a true cell. The erythrocyte lacks a nucleus. It lacks endoplasmic reticulum. It lacks mitochondria. It lacks ribosomes. It's basically a bag of hemoglobin. So all of these cells, whether we're talking about the cells of the immune system or the erythrocytes or the platelets, all of those formed elements have their origins in the red bone marrow. They all originate as proper cells in the red bone marrow. But as part of the development of these cells, particularly the erythrocytes are going to lose their nuclei and ribosomes and mitochondria. They're getting rid of all of those important cell functioning pieces of machinery in exchange for filling that plasma membrane full of hemoglobin, because its function here is going to be to carry oxygen. We're going to see it, particularly when we get to the end of the week, talk about respiration. We're going to be using these erythrocytes as the major player of taking on oxygen in the lung and giving up oxygen in the tissues, where additionally those erythrocytes in some cases will take on some carbon dioxide and transport it back to the lung. But one of our first protocols here is going to be simply describe all of these components. And again, we'll make some lists. And I'll try to give you some idea of the functions as we go through them. And one of the ideas that we'll come to is that erythrocytes are 45% of the blood volume. So they're a really important aspect of the blood. And then we have to think about this idea that if erythrocyte isn't a true cell, if it can't make its own proteins, then it must have a lifespan. So there's going to be sort of origin and fate of these erythrocytes. The erythrocytes are going to be born in the red bone marrow. They're going to be born from a cell type called a proerythrocyte. And the proerythrocyte will develop, and we'll go through some of the stages, and we'll end up with something known as a reticulocyte. The reticulocyte is the first cell of that lineage, which is going to leave the red bone marrow and go out into the blood vascular compartment. And it will then mature in the vasculature. It will mature into a proper erythrocyte. But we're going to see that we're not just generating these erythrocytes willy-nilly. They're under the control of a hormone, and they're under the control of a hormone called erythropoietin. Erythropoietin is generated by the kidney. Now, it seems strange that we'd be generating erythropoietin in the kidney, but it turns out the kidney has a tremendous blood supply. Again, in the next section, we'll talk in detail about this. But of the blood leaving the left ventricle, something like 20 to 25% of that blood is going to flow through the kidneys. There's a tremendous blood supply into the kidneys, and so the kidney now has developed a kind of mechanism to monitor the oxygen concentration in blood. As the oxygen concentration in blood begins to drop, as it begins to fall, then those cells, called pericytes, will then begin to produce the hormone erythropoietin. Erythropoietin is a peptide hormone. It's going to bind to receptors on the membranes of proerythrocytes in the red bone marrow. So the erythropoietin is produced in the kidney, but now goes into the blood, goes to the red bone marrow, and in the red bone marrow, it binds to these developing erythrocytes and signals an increase in division, an increase in cell survival, and thereby increases the number of erythrocytes. And we'll talk a little bit about how that happens. But just as there's an origin in these erythrocytes, because they've lost all their protein synthesis machinery, there's also a fate. And we'll see that the erythrocytes meet their end, for the most part, in the spleen. Now, in those people who lack a spleen, that function is taken over by the liver. But for the most part, we'll see for our purposes here that the worn out erythrocytes, the ones that are 3 to 4 months old, those will be recognized by macrophages in the spleen. And then those macrophages will basically endocytose the erythrocytes and destroy them, salvage some of the parts, and get rid of bits and pieces that they can't really salvage and recycle. And we'll talk about that. Now, one of the ideas, though, about the erythrocyte we have to spend some time on is their structure. They're really small cells. They're only about 2 to 3 micrometers in diameter and about 7 to 8 micrometers wide. And that small size dictates their function. Their function is to carry oxygen bound to hemoglobin. But for that hemoglobin to bind to the oxygen, we have to have very, very short diffusion pathways. One of the ideas we'll run into by Friday is that as the erythrocytes are in the pulmonary capillary beds, they have less than about 1/3 of a second to bind to oxygen, saturate all the binding sites of the hemoglobin, leave the lung, and go out into the rest of the vasculature. So that means we have very, very short periods of time in which to load the oxygen onto the hemoglobin, which means we need very short diffusion pathways. There's nothing magical about the loading of the oxygen onto the hemoglobin molecule. It's just by concentration gradients of oxygen and the diffusion pathway. Because of that, then diffusion is the rate limiting step. So what we do is we make these erythrocytes very skinny so that the oxygen can easily diffuse into them. And we'll talk in some detail about that. Then we're going to talk about what's in the erythrocytes, because this is going to be an important aspect of respiration. And so we'll see that the erythrocyte contains something in the neighborhood of about 250 million molecules of hemoglobin. And we'll see that each of those hemoglobin molecules is composed of 4 proteins, 2 alpha chains and 2 beta chains. Each alpha chain and each beta chain contains an iron. And that iron embedded in a porphyrin ring is now going to be the site to which oxygen binds. The porphyrin ring is this interesting structure where the electrons are being shared. And because the electrons are being shared, they're borrowing the electrons from the iron atom, leaving it slightly positively charged. That now allows the oxygen to bind to the iron atom. Now, the beauty of that is where oxygen is in extremely high concentrations, such as in the lung, the tendency is to bind the oxygen to the iron in the hemoglobin. But out in the tissues, where we're trying to deliver this oxygen, as the oxygen concentrations drop, because the oxygen is being taken up by the cells, under those circumstances, then the iron is more likely to relinquish the oxygen. So it has higher affinity in the lung than it has in the tissues. And we'll walk our way through that. But let's start with an image here. And this is an image that has happened to all of us. We go to the doctor's office, the phlebotomist comes out, and they stick a needle in our arm, and they basically take an aliquot of blood. And this blood in a tube is placed in a centrifuge. And the tube with the blood is spun in the centrifuge. And the spinning in the centrifuge separates the blood into three major components. So by spinning it, what we're going to do is we're going to force the densest portions of the blood to the bottom of the tube. So those things which are densest are going to move to the bottom of the tube. Those things which are least dense are going to move to the top of the tube. And when we do that, we find that the blood separates out into these components down here called hematocrit. And a region above called plasma. And a very narrow band in between the plasma and the hematocrit, known as the buffy coat. Now the players in each of these layers are that the hematocrit is composed of erythrocytes. Often you'll see this referred to as red blood cells. I don't like the term red blood cells. I'll often use it, but I don't like it very much because we're going to see that these are not true cells. They're shadows of real cells. They're basically bags of hemoglobin, the hemoglobin contained within the plasma membrane. So this is a specialized cell type, the erythrocyte. But when we look at the overall volume, we see that, in the neighborhood of about 45% of the volume is hematocrit. This will vary from individual to individual, but typically it's between about 42 and 45%. Then up here in the upper portion, the plasma is basically a water solution of proteins. The plasma is about 55% of the volume. And don't remember these numbers, but I want to tell you these numbers here to say that the water solution is about 90% water. It's about 8% protein and 2% ions and other stuff. So 98% of it is a protein solution in water. That's what the plasma is. And the reason that we see these two separate from one another is that each of these erythrocytes is going to contain iron. And because those erythrocytes contain iron, this iron which is much heavier than the nitrogen and carbon and hydrogen and oxygen or the rest of the amino acids in the hemoglobin, that makes this molecule of hemoglobin more dense. So iron makes hemoglobin denser than the other components. Of blood. It's that increased density that allows for the blood to be spun to the bottom of the tube. It's that increased density under the effects of the centrifuge, which removes the erythrocytes to the bottom of the tube and separates the blood into the hematocrit, the buffy coat, and the plasma. Now the plasma is just proteins. And the next question is, what's in the buffy coat? Well, the buffy coat contains cells of the immune system, and another structure called a platelet. The platelet is not a true cell. So we give it this euphemism of a formed element. Also, the erythrocytes, because the erythrocytes lack nuclei and mitochondria, these are also formed elements. The only components of the blood that are true cells, proper cells, have all the cell machinery you'd associate with a cell, are the cells of immunity. These are, in fact, true cells. And we'll list these here in a little bit. So what we've done here is we've simply separated the blood into its different components. About 45% is the hematocrit, about 55% is the plasma, and the buffy coat is in fact less than 1%. It's a very, very small part of the volume of the blood. The majority of it is just this sort of water solution with protein. About 45% is the hematocrit, which are erythrocytes, and then we have this tiny sliver of less than 1% being the buffy coat. So let's now drill down a little bit and talk where these cells are coming from. And spell out the idea here that all cells in the blood originate as hematopoietic stem cells, sometimes you see these referred to as hemocytoblasts. In the red bone marrow. And it doesn't matter whether we're talking about the buffy coat, where we have macrophages, we have granulocytes, we have lymphocytes. These are going to be, I'll put this in parentheses, this is going to be both B lymphocytes and T lymphocytes. And one more character, which are called the platelets. See if I'm not missing. Yeah. The platelets, and we're not going to spend much time on these, but the platelets are not true cells. They are basically plasma membranes surrounding a bunch of vesicles. And these vesicles contain serotonin and other clotting agents. The source of these platelets, let me do this here, source of platelets in the red marrow is a cell type called the megakaryocytes. The megakaryocytes, as the name implies, are these great big cells. Often they're even multinucleate. The megakaryocytes reside in the red bone marrow, and they'll reside right next door to a capillary. What I'm drawing in green is meant to be the endothelium of a capillary in the red marrow. And what we'll see is that outside the capillaries, there will be this cell called a megakaryocyte. The megakaryocyte sends out a process, basically an extension of its cytoplasm. That extension of its cytoplasm surrounded by plasma membrane insinuates between adjacent endothelial cells. And then this bit breaks off. And when it breaks off, it contains the components of the cytoplasm that were in this extension. And this now is a platelet. I don't want to get too deeply into this. It turns out that platelets are essential for blood clotting. That's about all I'll say about it. Now, if you get into the blood clotting story, and I don't mean to belittle or demean the importance of blood clotting, it's essential for us in our lives. But if you get into the details of it, there are something like 30 different proteins involved. And they're numbered in the order in which they were discovered, not in the order in which they function. And so you end up with this long list of confusing proteins, and I just have I don't want to think about it, and I don't want you to think about it either. So we're going to limit our discussion of clotting. If you take another course in hematology or in physiology, you may learn in much more detail about this. It's really important. But let me tell you what the platelets do. The platelets initiate blood clotting. Now, the function of the platelets in blood clotting is pretty clear. When you cut yourself, you start to bleed, you need to stop that bleeding. And these platelets are one of the first formed elements, one of the first structures involved in the clotting. If you don't have adequate platelets, you can't clot blood. And that's certainly a big problem if you cut yourself. But it's also a problem in day-to-day living, because all the time we'll have these small breaches in the capillaries, and we have to plug them. And the first thing that happens is we plug them with what they call platelet plug formation. And we plug that breach with the platelets. And when we do that, that's the first step in blood clotting. Yes?
So people with that condition where they can't clot their blood properly, like that one condition that Russian prince had, does this affect that? Or?
So the question is about hemophilia. Hemophilia is a disease that is a problem, and I don't remember which one of the clotting factors is deficient. But one of the clotting factors is not produced because these individuals have two bad copies of the gene for making that clotting factor. And so they can't properly clot their blood. And so they often will die at a fairly early age. We can control it much better now, but they often die at a very early age because of this inability to clot blood. The way we have fixed that problem is we know the clotting factors. And so what we'll do is we'll take blood from volunteers and then purify that blood and purify the clotting factors. And by purifying the clotting factors, then we can replace the clotting factors that are deficient in those individuals with hemophilia. In the early days of HIV, one of the large populations of individuals who are coming down with HIV and then AIDS were hemophiliacs, because in the process of purifying the blood and purifying the the component that these individuals needed for blood clotting, along came the virus. And so with the drugs that people were using to replace the missing clotting factors, they were also taking in HIV. And so you had a very high incidence in the, before we understood a lot that, in the 80s and early 90s, you had a fairly high incidence of hemophilia, of HIV among hemophiliacs, because they got it from tainted blood. But all of that is to say that that is one of the clotting factors. If you talk to hematologists, there are a whole raft of these mutations that cause deficiencies in the chain of proteins for blood clotting. There's some people who it's Leydig factor 5, and they lack something called Leydig factor 5. And that's an important regulator in the production of blood clots. And these individuals throw too many of them. They clot too easily. And so those individuals tend to be on a continual regimen of what they call blood thinners, usually something like Coumadin or warfarin, to keep the blood clotting from happening. And the other thing is that these platelets, if you lose platelets, what will sometimes happen is you'll die of hemorrhaging and then a series of blood clots. And the story I want to tell you here is there is a common problem, and this is a problem that emergency room physicians, and particularly intensive care physicians, have to deal with. And that is that often in people who are immune suppressed, they'll get a bacterial infection. Then the bacterial infection, those bacteria produce these exotoxins, which then begin to activate the platelets. And the platelets then begin to degranulate. They lose their vesicles. And then you start throwing these tiny clots all over the body. And this is a problem because these tiny clots, these tiny emboli, are going to stick in the lungs, the capillaries of the lungs, the brain, the heart. And so now you have all of these blockages throughout the blood vascular system. So the first thing you think of is, well, what we'll do is we'll give them some kind of treatment that will dissolve those clots. Something like Coumadin, which is warfarin, or none of the others. But the problem is when you do that and you when you start to destroy those clots, those emboli, because these individuals have used up all of the clotting factors, they can't clot in any way now. So the physician is really on the horns of a dilemma. Do you treat the clots, the emboli, or do you treat the fact that these people can't generate new platelet plugs and plug up breaks in their vascular system? So you're on this very narrow tightrope trying to decide whether or not you're going to treat the clot produced by the bacterium, or whether you're going to treat the hemorrhagic problems. And it's a very difficult problem. You have to balance it just right. And often they don't. And in fact, the term here is disseminated intravascular coagulation. Disseminated intravascular coagulation is commonly abbreviated as DIC, and the dark humor here, the gallows humor, is that stands for death is coming, because it's very difficult to control both the generation of the emboli and because the clotting factors have been exhausted to then prevent the hemorrhagic problems that may ensue. But all of that is to simply sum up to say we really, really need these platelets, and we need all these clotting factors to regulate this cascade of events, to properly regulate the function of the blood in terms of forming blood clots. But these are not true cells. They are formed elements. They're shadows of real cells. They're basically a plasma membrane with some vesicles around them. But the other character down here in the hematocrit, which is also not a true cell, is the erythrocyte. And what I want to emphasize here is the lineage of these erythrocytes, and emphasize the fact that these are really not true cells. So take this panel and label the panel as erythrocyte development, and recognize as we go through this that one of the hallmarks of erythrocyte development is all of the components of the cell that the erythrocyte loses. So if we take this panel and talk about erythrocyte development, in the red marrow. We'll start out with a cell that has a nucleus, and this is called an hematopoietic stem cell. This hematopoietic stem cell, given the right growth factors, the right set of cytokines, can develop into any of those cells of the blood system. It could develop into a macrophage, it could develop into a granulocyte, it could develop into a lymphocyte. It can go any direction. But for our purposes, we want to think about the series of events. And if you take a course in hematology, you'll learn all about the growth factors. But there are a series of growth factors which will guide this cell to developing into something known as a pro-erythrocyte. Now, once this hematopoietic stem cell, which is in fact totipotent, meaning it can go in any direction, once the growth factors have forced the cell to develop into a pro-erythrocyte, it's on the road to making erythrocytes. It can't be turned back. But this pro-erythrocyte, again, in the red marrow, all of this is in the red marrow. This pro-erythrocyte develops into another cell type called a normoblast. Now, if you look up the details of erythropoiesis, which we should say, we can give it a formal name of erythropoiesis. If you look up the details of erythropoiesis, you'll see there are a number of named cell types in between. They're named by the kinds of dyes they take up. But what I want you to recognize is we start out with an hematopoietic stem cell. We develop into a pro-erythrocyte. And then we develop into something called a normoblast. The why I want you to know about the normoblast is what happens in the normoblast is in the normoblast stage, the nucleus is ejected. That's a pretty serious thing to do because, of course, the nucleus contains all of the genetic machinery for making RNA, and it contains all the genetic information of how you make any protein. And so once you've gotten rid of the nucleus, this cell can no longer make new proteins. But in addition to getting rid of the nucleus, we also get rid of mitochondria. And we also get rid of ribosomes. And we get rid of most of the endoplasmic reticulum. Now again, this normoblast is found in the red marrow. And the normoblast will develop into another cell type called a reticulocyte. It derives its name of reticulocyte because it has remnants of endoplasmic reticulum. So you get these faint pieces of ER. But it's not organized, it's bits and pieces of ER. Now, at this point, you can draw a line. Everything to the left of the line is in the red marrow. And to the right of the line, these reticulocytes now enter the blood vascular compartment. That is to say that these erythrocytes, these reticulocytes leave the red bone marrow and go out into the blood vascular compartment, and they will now be a bag of hemoglobin that will develop into an erythrocyte. In the blood vascular compartment. I think you can see that once you have a cell that's gotten rid of all its protein synthesis machinery, that cell can't proliferate. It can't make copies of itself, and it therefore has a lifespan. And these erythrocytes have a lifespan of about 90 to 120 days. So once these erythrocytes are out and about, they only last for 3 or 4 months. And we have so many of these that every minute of every day we're producing tens of millions of these erythrocytes. As you sit here in this room, every minute you're producing several million of these erythrocytes to replace the ones that are worn out, because there's this constant turnover of these erythrocytes. And so that brings up the question of a little bit of the control. Let's go to the next panel and talk about the fate of the erythrocytes. And go up here and talk about erythrocyte fate. So if they're born in the red bone marrow, these erythrocytes are then going to meet their end in another structure, predominantly the spleen. Now, I'll say that in those people who've had splenectomies, and that's not uncommon, the function of the erythrocyte destruction is taken over from the spleen by the liver. But for the most part, the worn out erythrocytes, I can just say here that the worn out erythrocytes are destroyed. In the spleen. Well, it's worth taking a minute here and talking about what the characteristics of the spleen are that allow for this function. The spleen is heavily vascularized. The spleen contains a splenic artery, and of course it's drained by a splenic vein. As with all of the vascular system we've talked about, this splenic artery will branch into arterioles and eventually branch into capillaries. And the capillary blood will pass through the capillaries and then pass back out through the splenic vein. But I want to spend a moment on the splenic capillaries. And we'll say that the splenic capillaries are fenestrated. Fenestrated comes from the Latin, it means window. Now, in reality, what this means for the splenic capillaries is that there are large gaps between adjacent endothelial cells. Because of these large gaps between adjacent endothelial cells, there's room for erythrocytes to leave the blood vascular compartment and get out into the extracellular environment, the extracellular space in the spleen. There will be regions in here, in the spleen, called the red pulp. And the red pulp is the site where erythrocytes and macrophages encounter one another. Now remember that the macrophages are endocytic cells. So if we draw one of these macrophages, what the macrophage will do is the macrophage recognizes worn out erythrocytes by the oxidized sugars on the erythrocyte. So again, if you're an erythrocyte, the transmembrane proteins that you were born with in the red bone marrow, those transmembrane proteins, of course, are glycosylated. These are meant to be sugars on proteins. We should probably say membrane proteins. These sugars, because they haven't been replaced, those sugars, as they're wandering around on the erythrocytes in the blood vascular compartment over a period of time of 3 to 4 months, those sugars now will become oxidized. They change their structure. That change in structure now can be recognized by proteins on the erythrocyte. So now the worn out erythrocyte becomes bound to the macrophage, and then through the process of endocytosis, the erythrocyte is brought into the cell, packaged into vesicles, and sent off to the lysosome. Now, of course, in the lysosome, the erythrocyte is going to be basically digested into its component parts. The amino acids will be recycled. We'll see here in a little bit an important atom in the hemoglobin molecule is an iron. That iron is going to be recycled. But there's something called a porphyrin ring. It's the porphyrin ring where the iron resides in hemoglobin. that is not recyclable. It becomes a waste product, and this porphyrin ring becomes a waste product that goes out as bilirubin and biliverdin. Now, the importance of this actually is mostly clinical. If you are in liver failure, what will happen is that you cease to be able to produce bilirubin and biliverdin and get rid of it. And so often if you see people who are in liver failure, they become jaundiced. They become quite yellow. And that's because the bilirubin and biliverdin is a yellow color. It's kind of a yellow-green color. Sometimes it'll be referred to as a kind of a bilious color. And that's where the jaundice is coming from. Now, another place where you see commonly this so-called jaundice is in newborn babies. And the reason for that is that when you're in utero, when you're living in utero, all of the oxygen you're getting to use is coming from your mother. So your mother is loading up her erythrocytes with oxygen in her lungs. That blood then is being circulated to the placenta, and then across the placenta, that oxygen is exchanged. And if you think about it for a little bit, you'll recognize that the fetal hemoglobin needs to have a higher affinity for oxygen than your mother's hemoglobin, because you need to be taking that hemoglobin, you need to be taking that oxygen from your mother's hemoglobin. So fetal hemoglobin isn't the same thing as adult hemoglobin. We'll see, it has a slightly different construction. We'll talk about that in a minute. And what's going to happen is that the hemoglobin in these fetuses at the time of birth has to change over within the first few weeks. Has to change over from the high affinity fetal hemoglobin to the normal hemoglobin. And so during that period of time, you have a tremendous destruction of erythrocytes. And as these erythrocytes are being destroyed in the process we described here, there is a byproduct from the hemoglobin molecule, which you can't digest or get rid of, and these are the porphyrin rings, which generate bilirubin and biliverdin. And if the baby isn't very good at getting rid of these molecules, they become jaundiced. It's an easy solution now. We put the babies under ultraviolet lights, and the ultraviolet light destroys the bilirubin and biliverdin. There was a time when the advice was, well, take that baby out and put it in the sunshine. And that worked too, because it broke down the porphyrin rings. All of that is simply to say that these porphyrin rings are a waste product that we can't really easily recycle when we're destroying the erythrocytes. And so we get rid of it through the liver as a waste product. But just as there is a fate of those erythrocytes, there's also, let's do this and we'll come back. There's also control of the origin. And the control of the origin of the erythrocytes, the control in the red bone marrow, is by hormones. So we'll take this panel and we'll label it as erythropoietin regulates erythrocyte production. Erythropoietin regulates erythrocyte production. Now it's odd, but erythropoietin is synthesized in the kidney. And the story here is that the renal artery provides blood to the kidney. We're going to go into great detail on this in a couple of weeks. But the point I want to make is that something like 20 to 25% of the blood leaving the left ventricle passes through the kidney. So there's this tremendous blood supply to the kidney, and there are cells in the kidney called pericytes. Monitor the oxygen concentration in blood. And in response to falling oxygen concentration, these pericytes secrete the peptide hormone. Erythropoietin. Erythropoietin, as a peptide hormone, binds to receptors on proerythrocytes. Remember, those proerythrocytes are the precursor cells in the red bone marrow for erythrocytes. So this is a peptide hormone that goes from the kidney to the red bone marrow, binds to receptors, transmembrane protein receptors in those proerythrocytes, and signals those proerythrocytes, probably to some extent, to make additional growth factors. It increases the rate of cell division, and it increases the rate of cell survival among the proerythrocytes. By increasing the number of proerythrocytes that survive, you now increase the number of erythrocytes produced. So this is a control mechanism for regulating hematocrit. It regulates the number of erythrocytes. Let's take a break, and I'll come back and tell you the story of erythropoietin and how it has a sort of an athletic history. Again. So let me tell you the story of EPO. Erythropoietin is often abbreviated EPO. And it's a peptide hormone, so it only contains a few amino acids. And it was realized probably, half a century ago now, 50 years ago, that people who are on kidney dialysis had very low hematocrits. And the reason for that is when you're on kidney dialysis, you're in kidney failure. And so when the kidneys no longer function, then they don't produce erythropoietin. Because they don't produce erythropoietin, these individuals are typically anemic. And so it was recognized by the pharmaceutical companies that since this is a peptide hormone, that if you could find the gene in a human that produced the erythropoietin, that you could take that gene and then splice it into some sort of cell that would generate erythropoietin. And they chose a particular cell type called the Chinese hamster ovary cells. And the reason for that is that you can grow Chinese hamster ovary cells up in liquid culture, just in free cell medium. And by then inducing, or basically putting into these Chinese hamster ovary cells, putting this erythropoietin gene into their genome, you could then get these cells living in culture to generate tremendous amounts of erythropoietin. This was a boon to the pharmaceutical companies because now they had all these people who are on dialysis and they could make their lives a lot better by injecting them with erythropoietin. It's also used commonly for people who are undergoing chemotherapy because in chemotherapy, the function of the chemotherapeutic drugs is to slow down cell division. And you want to slow down cell division of the neoplastic cells in the tumor, but in that process you also slow down cell division in the red bone marrow. And so these people are often anemic because they can't generate enough proerythrocytes from their hematopoietic stem cells to generate enough erythrocytes. And so we also use erythropoietin to help people get through periods of chemotherapy when their erythrocyte counts, their hematocrits are low. But it was also recognized by athletes. And it was particularly recognized by athletes who were long-distance competitors, especially bicyclists. If you're involved with professional bicycling in Europe, you can make hundreds of thousands of dollars a year. But you have to be at the very top level of the competitions. And they began to recognize that these bicyclists are astonishing. They'll get on a bicycle in the morning and they'll pedal for 6 hours, and they'll be at very high aerobic consumption during all that time. And so they began to recognize, well, gee, I might be able to get a competitive edge if I could just inject some erythropoietin. And what's more is the erythropoietin is from a human gene, and so the protein is nearly identical to what you would see produced by your own body. So your kidney produces that erythropoietin, but now you get a boost. You inject some. And after every race, the top finishers have blood collected, and the blood is then checked for various kinds of performance-enhancing drugs. But they couldn't check for the erythropoietin because it's a protein. And it's a protein that's a gene product of humans. So how are they going to distinguish it? And for years, these bicyclists said, they'll never catch us because of the characteristics of the erythropoietin. But they still kept those vials of blood frozen back for years and years and years. And so then someone discovered that the pattern of glycosylation, the way the sugars are put on the erythropoietin in the Chinese hamster ovary cell, is just a little bit different than the way it's put on proteins in, when it's put onto proteins in the human kidney. And so there was a subtle distinction between the sugar patterns, the glycosylation patterns in the erythropoietins. And that's how they went back and caught people. They went back and looked at the pattern of glycosylation on the erythropoietin. And if it looked like Chinese hamster ovary cell erythropoietin, then they were busted. But if it were human, then of course they were fine. But that's how Lance Armstrong got caught. He got caught because if he'd been a better cell biologist, he would have understood that the erythropoietin was different. But he didn't know at the time, and I don't think anyone knew at the time. But that's basically what busted a lot of those high performance bicyclists. The other story I want to tell you, tell you here before we get into the structure of the erythrocyte, is back here at this site of erythrocyte destruction. Because erythrocytes, yes?
Question about that bicyclist story. Wasn't it also blooding, like where they would take blood out of their system and then put it back in?
Oh, okay. So this is a different question. The question is, what's the difference between erythropoietin and blood packing? Yeah. And they would do blood packing, and the blood packing goes. The first time I heard of somebody doing this was, I think he was a Finnish marathon runner, and he was medically trained, and so he recognized that you could take blood and freeze it back and then have transfusions. So he would take blood and freeze it back, and then just before a race, he would give himself a transfusion. So that would elevate his erythrocyte count, elevate his hematocrit, he could carry more oxygen. And it worked pretty well. And they still do that. And that's a hard one to catch because it's your own blood. And so it's very difficult to distinguish blood which has been taken from your own body, frozen back, and then put back again to raise the hematocrit. One of the tests was to simply sample people's hematocrit. And if they had hematocrit above a certain point, it was assumed that they were cheating in some way. But that never seemed fair to me because these athletes are incredible anyway. I mean, so it wouldn't be so surprising that they would have high hematocrits to be able to sustain that kind of aerobic function. So what's the difference between selecting people who are just good at this and have the physiology for it and those who are injecting themselves with performance-enhancing drugs? That's a hard thing to catch with the blood packing. But the other story I want to tell you was the one about the destruction of erythrocytes. And because the erythrocytes have a 90 to 120 day lifespan. One of the things that happens in erythrocytes, if you live in a part of the world where malaria is prevalent, is that there's a parasite called a plasmodium. It's a eukaryotic parasite, and the plasmodium of malaria gets into the erythrocytes. And this disease is really seriously a problem in areas where you have high instances of malaria. There are places in West Africa where, until the last few years, where we were better at mosquito control, you would find 1 in 3, 1 in 4 children would die before the age of 5 from malaria. Similarly, if you looked around the Mediterranean area, 1 in 3 or 1 in 4 children in Sicily and parts of Greece would die before they reached 4 or 5 years old, and they died because of malaria. Well, biology had a fix for this. What biology did is it changed the hemoglobin structure. In West Africa, it was a single site mutation, which led to a single amino acid substitution. But this changed the characteristics of the hemoglobin. And if you had one copy of the gene that made the new or mutated, let's say it's mutated, protein, what that would do is it would shorten the lifespan of the erythrocytes. Well, these plasmodia tend to require 90 to 120 days to mature. So by turning over the erythrocytes more rapidly than the lifespan of the plasmodium, these individuals were protected against malaria. And the single site mutation in Africa, in West Africa, is called the sickle cell trait. If you only have one bad copy, you can live pretty normal life. But if you have two bad copies, two mutated genes, then you actually get sickle cell anemia. And what happens if you look at those cells, rather than having this particular characteristic shape, they end up looking like half moons or sickles. These then tend to stick in the capillaries. So if you have two bad copies, these children typically don't live beyond the age of 4 or 5. But if you have one good copy and one bad copy, you can lead a normal life and you're protected against malaria. In parts of Africa, and parts of West Africa, where malaria is endemic, something like 10% of the population, 10 to 12% of the population carries this gene. That means when two people have children with a background of 10%, it's very often that you have two individuals who have a sickle cell trait. That means 1 in 4 of their children will be sickle cell. 1 in half of their children will carry the trait and be able to not die of malaria, and half their children, or 1/4 of their children, are normal. But the problem is you see a high percentage of these children having sickle cell anemia. And even in the United States, because of the horrors of the slave trade, because of that slave trade, and most of those individuals originally came from West Africa, even in the African American population in the United States to this day, there's something like 12% of the population, no, I think it's around 8%. 8% of the African American population carries this trait. So it's a very common problem in the African American community. Now, the Europeans solved this problem in a different way. And in Europe, the problem is something called thalassemia, TH. And thalassemia, rather than being a single site mutation, is a deletion in the hemoglobin gene. But again, you have a bad copy, because you have one good copy and one bad copy. That shortens the lifespan of the erythrocytes. And the same story pertains, where now the plasmodium can't live out its full lifespan. And it brings us to an interesting question, I think, in that there's no question that we should be treating individuals who have sickle cell anemia and making their lives as the best we can. But there's a tendency to say, well, this is a bad gene in the population. Let's get rid of that gene. Let's find ways to eliminate the gene. Let's do basically gene surgery. Take that bad gene out and replace it with a good gene. But there's a real ethical question about that, because if you replace that gene in the germline, now you have removed a part of the human population who are no longer susceptible, who are effectively immune to sickle cell anemia. Do you want to remove that gene from the population? I think that's a really difficult ethical question, because for the individual to have two bad copies is deadly. And for those people who carry it, there's always a worry that you're going to have children with someone who has the same trait. And 1 in 4 of your children can carry this horrible disease. But for the population, it may be better to leave that gene in the population. So you've got this really interesting ethical dilemma that we're going to face in the next few years about what do we do with these genes that seem to impart some selective advantage historically, but now don't seem to be that much of a selective advantage, and we would be happy to get rid of them for those individuals. I think that's a really interesting ethical question that we're going to have to wrestle with over the next 50 to 75 years. Right now, the ethical stand is don't mess with the genome, because we just don't know which of those genes that are out there have some kind of selective advantage. And if we get rid of them, that may be bad for the overall population. So that's the thinking right now. But with CRISPR, we'll be able to do that. And so it's going to become an issue. So any questions up to that? Yes?
Could you clarify why having the single site mutation is not detrimental to health, but having the double mutated gene causes
Okay. So it turns out, if we look at, so the question here is, why is it the one bad copy isn't a problem, but two bad copies is? So if, and again, this will be something that might be a little bit clearer in a few minutes here when we get to the proteins in hemoglobin. Turns out hemoglobin contains 4 proteins. It contains 2 alphas, this way, and 2 betas. The mutation is in the beta chain. It's for the gene for the beta chain. There don't seem to be very many mutations in the alphas. And I think it's just too deleterious. Whereas you get these mutations in the beta chain, and now when you have these 4 hemoglobin molecules, 2 betas and 2 alphas, these assemble randomly. And so in some of your proteins, well, some of your hemoglobin proteins, you'll have 1 good copy and 1 bad copy. Now it's true, in some of them you'll have 2 bad copies, and some of them have 2 good copies. But once you begin getting these mixtures of good copies and bad copies, then for reasons I don't quite understand, those erythrocytes are recognized as being somewhat deficient. They seem to be wearing out more quickly. And because of that, they're recognized as worn out more quickly, so they're taken out of the circulation more quickly. So that means you still have enough good hemoglobin molecules in here to carry enough oxygen to supply your tissues. So it's not so deficient that you can't, I'll be with you in a minute. It's not so deficient that you can't deliver enough oxygen to your tissues, but it is deficient enough that the system will see it as a deficient erythrocyte that has to be destroyed more quickly. So rather than 90 to 120 days, maybe it lasts 60 to 90. But because of that shorter lifespan of the erythrocyte, then the plasmodium doesn't have a chance to go through its full life cycle. But since you have enough good copies of the hemoglobin that offset the number of bad copies, then you can live just fine. But when all of your alphas now are mutated, then these don't bind oxygen properly. And what's worse is they don't maintain this nice, neat, what they call this dumbbell shape. And instead, they'll shift under, particularly when low concentrations of oxygen, they shift into this sickle. And then these get stuck in the capillaries. Did that answer your question?
Yes, so just to clarify real quick, so essentially if you have a single site mutation, it's enough to be beneficial but not so detrimental to where it's affecting the overall structure?
That's right.
Okay.
I mean, if you look at athletes, they may not be long distance runners that carry the sickle cell trait, but they can be astonishing athletes. For instance, the number of players in the National Football League who carry the sickle cell trait is quite high. So they don't seem to be compromised in their athletic abilities. They probably aren't going to be marathon runners, but they can be outstanding athletes with this and do quite aerobic kinds of things. So you don't lose that much function with these mutations. And it's true with the thalassemia too. You get bad copies that aren't much good to you for that beta subunit. No, they should be, the betas are the ones that are bad, so not the alphas. And that's true in beta thalassemia too. Beta tells you which protein it is, and that's a deletion. And those people then have, if they have good copies and bad copies, they do all right. If they have 2 bad copies, then they're in trouble. Okay. You had a question in the back.
Yeah, my question was though that thalassemia. So is there a change in the shape of the blood cell or no?
I don't know that the thalassemia changes the shape of the blood cell to sickle like the sickle cell does. I don't know that you see that as well. You'd have to talk to a hematologist about that. I don't know if it's recognizable in that way, or if there are other tests. It took a while to sort that out, because they first thought that the issues of individuals in the Mediterranean region were going to be the same as in West Africa, and it turns out it's a different mutation. It's a different way to solve the problem. But both of them are focused. It's kind of convergent evolution on shortening the lifespan of the erythrocytes so that the plasmodia can't go through a full life cycle. But I don't know that if there are ways that a blood smear you can see it. Yes?
When the erythrocyte's developing in the marrow and it ejects the organelles, do the organelles get recycled?
So what happens to the organelles? As far as I know, those organelles are gobbled up by macrophages. I don't know of any mechanism that could take the nucleus or a ribosome and say, oh, this is in the wrong spot, let's tuck it back into this cell. I don't think that happens. I think that it's just destroyed. Anything else? Okay, let's now, since I've skated around it, let's now begin talking a little bit about erythrocyte structure and why it's important. The erythrocyte has this classic structure called a biconcave disc. And as we alluded to here with talking about the structure of the sickle cell, the sickled erythrocytes, they need to have this particular shape to do their job. So I want to think a little bit about why that is. And when we look at these erythrocytes, they look a little bit like this. They're said to be a biconcave disc. At their widest point here, they're only about 2 to 3 micrometers wide. And the diameter of these things is about 7 to 8 micrometers. So they're pretty small cells. And they're small, the small size optimizes short diffusion distances. We're going to talk about this in a lot more detail toward the end of the week, but I just want to point out now that I'm going to draw here something known as an alveolus, or an air sac. Right next door to the epithelial cells of the alveolus is a pulmonary capillary. Now the energy is going to be provided by the right ventricle, but the erythrocytes are going to be lined up in single file, and I'm going to show it here, moving left to right, past the alveolus. The time it takes, even when you're sitting here quietly, the time it takes for an erythrocyte to pass by one of these air sacs is in the neighborhood of about 0.75 seconds. When you're exercising hard, when you're really working out, these numbers get down to about 0.25 seconds. The only way you can load these erythrocytes with oxygen is by diffusion. The oxygen has to diffuse out of the air into the erythrocytes, bind to organoids, the hemoglobin in the erythrocytes, and fully saturate those hemoglobin molecules. That has to happen in less than 0.7 of a second. For this to work, you must have short diffusion pathways. You simply can't have the oxygen moving over great distances. And in fact, if you begin to fill this alveolar sac with fluid, you increase the diffusion distances to such a point you can't adequately oxygenate the hemoglobin. That's pneumonia. Well, under these circumstances, we need very short diffusion distances so the oxygen can diffuse into the erythrocyte where it binds to hemoglobin. The hemoglobin is the functional reason to have erythrocytes. And the reason those erythrocytes have gotten rid of all of their other organelles, those erythrocytes have now become basically a bag of hemoglobin. In the process of ejecting the nucleus and the mitochondria, you end up with this cell, this tiny cell, that has 250 million hemoglobin molecules. Each hemoglobin molecule contains 4 oxygen binding sites. So each one of these erythrocytes can bind 1 billion. Let me do it this way. 10 to the 9th, 1 billion molecules of oxygen. So what we have produced in this process of generating an erythrocyte, getting rid of the nucleus, getting rid of the mitochondria, getting rid of all the protein synthesis machinery, we now basically have a plasma membrane that's surrounding a bunch of hemoglobin molecules. That's its function. And its function now is to use the hemoglobin to bind to oxygen in the lung. So let's now look a little bit at the structure of the hemoglobin to see if we can think about how that's working. So any questions up to there? So now I'm going to draw the 4 protein molecules as kind of squiggles, but recognize they have a proper 3-dimensional structure, and that proper 3-dimensional structure, as the sickle cell anemia implies, is really important for the shape of the erythrocyte as well as the shape of the hemoglobin molecule. But I'm going to simplify this and say that each hemoglobin contains 4 proteins. Among these are 2 alpha chains and 2 beta chains. Turns out there's an alpha gene, actually there are 2 alpha genes and they're in tandem, they're one right behind the other, and there's a beta gene. Now, when we look at these, let's do it this way. We have these 4 proteins. I'm just draw them as squiggles. Remember, these are in 3-dimensional shapes. And these are going to be alpha and alpha. And then there'll be these two beta chains. They're very similar in structure. And so these are two beta chains. Now, if we look carefully in each one of these, there's a site within the protein where we have bound an iron atom. So each one of these 4 individual proteins, the two betas and 2 alphas, each has an iron atom. And the iron, the Fe, is the site where oxygen binds to hemoglobin. And if we look carefully at this, we find that within the protein, the iron is complexed in a structure. And I'll draw it for you. I'll never ask you to repeat this, but where there are 4 nitrogens, and the 4 nitrogens are each in a 5-membered ring. Then each of these rings are further connected by ring structures, so that the electron sharing in the rings permits the movement of electrons in the iron. So that when these electrons are borrowed, because the electrons orbiting around the iron can be moved by this structure, this ends up taking on a positive charge. The positive charge in the iron now can bind to oxygen. But the beauty of this system is that oxygen binds to the iron in this structure known as the porphyrin ring. So the oxygen binds to the iron in the porphyrin ring when it's in the lungs up here. Up here in the lungs, there's a tendency for the oxygen to bind to the iron. Binds loosely, but it binds. Then when these erythrocytes leave the pulmonary capillaries, go back to the left side of the heart, get pumped out into the systemic vasculature, out in the systemic vasculature where the oxygen levels are much lower than they were in the lung, then these porphyrin rings, the iron in the porphyrin rings, relinquishes the oxygen. So it's a wonderful system, such that in the lungs, oxygen binds to hemoglobin, binds to the iron in hemoglobin. But in the tissues, where oxygen levels are lower, then the loose affinity between the oxygen and the iron allows the oxygen to leave and go into the tissues. So it's this wonderful molecule which, in regions of high oxygen concentration, takes on oxygen. And in regions of low oxygen concentration, it relinquishes that oxygen so that that oxygen can be taken up by the local cells and used in the mitochondria. So it's this great structure. Now, if you take a biochemistry course, you'll find that you can vary the structure in here, and that will change the affinity of the iron as well. Under certain anaerobic conditions, you'll get these iron atoms to be have lower affinity for oxygen and be more likely to give it up. But for our purposes, just remember that the oxygen is binding to the iron. And then it binds to the iron where in regions of high oxygen concentration, and it releases from the iron in regions of low oxygen concentration. And that'll be important when we start talking about the respiratory system. The last piece here is that this is the porphyrin ring that's waste. Let me show you a couple of slides. I think it's easiest to show this in some slides. So this is the structure of the erythrocyte itself. And so it simply shows that biconcave disc and represents the idea that these, at the edges, these cells are only 2 or 3 micrometers thick. And in the center here, in the biconcave region, that's even narrower. And all of that is useful for having short sorts of diffusion distances. This is a diagrammatic representation of the hemoglobin molecule. And so it's showing the 2 alpha chains and the 2 beta chains. And here they're showing this disc-like structure being the porphyrin ring. This is the full detail of the porphyrin ring. Again, I'll never ask you to repeat this, but note how the iron here is coordinated with the nitrogens in these 5-membered rings at 4 sites. And with all these electrons moving around in this ring structure out here, what you do is you now are going to borrow the electrons from the iron. And by borrowing the electrons from the iron, you will make it slightly positive. And by making it slightly positive, you now allow for the oxygen to bind to that iron. Yes?
You answer right there.
Okay. And so in the lung, because of this positive charge on the iron, the oxygen binds freely to it. But then out in the tissues, oxygen can leave the iron, and this then becomes, and then this relinquishes the oxygen, so the oxygen could be used in the tissues. The other idea here is this ring structure, this porphyrin ring. We can pop this iron out of the ring, and we do that. We'll remove, when we're recycling erythrocytes, we'll remove the iron, and then we'll store it in the liver. So we have sort of a readily available storage site of iron in the liver. But this rest of this structure, this ring, is not recyclable, and that's what becomes bilirubin and biliverdin. And because it has a kind of yellow-green color to it, that's the bilious nature and the yellow nature of jaundice, because these will then build up in the skin and cause damage. In fact, yellow fever is produced by a virus, and it's, for years, it's carried by a mosquito, and it was the bane of living in the tropics. In fact, it's one of the reasons the French never completed the Panama Canal, because of yellow fever. And until they could figure out how to control the mosquitoes, they couldn't finish the canal. But they figured out how to control the mosquitoes, they controlled the yellow fever, and they finished the canal. But the point here is yellow fever results because that virus destroys the liver. And when the liver is destroyed, then you no longer can get rid of these porphyrin rings. And you get the buildup of this waste product in the skin, and people become jaundiced. And that was the source of the term yellow fever. Well, any questions? Yeah?
So is it the iron, or I just missed that? Is it the iron that causes the, or a component, another component?
No, it isn't the iron. The iron doesn't cause the jaundice. Because we'll actually take the iron out of the ring. All we have left over is the ring. And the ring structure itself can't be further broken down. And so it's taken to the liver and goes out through the bile. And we'll see later that some parts of bile are actually useful in emulsifying fats, but this is just a waste product, and we get rid of it. But it has this characteristic yellow-green color. And because of that yellow-green color, when your liver is not getting rid of it properly, that porphyrin ring, without the iron, is building up under the skin. And that's true for the jaundice babies too. They're recycling all of that fetal hemoglobin, because the fetal hemoglobin isn't 2 alphas and 2 betas, it's 2 alphas and 2 gammas. And so those two other proteins in the fetal hemoglobin give the fetal hemoglobin a slightly higher affinity for oxygen than the adult hemoglobin. And that's the hemoglobin that's recycled shortly after birth if things are working properly. Yes?
In the in the places with low O2 concentration, what makes the O2 want to leave this iron, the hemoglobin?
It's just law of mass action. In regions where you have lots of oxygen, it binds to the iron. And then as the oxygen concentrations drop, then it comes off. That's the first level. There's some other levels of control with diphosphoglycerate and some other things which will affect the shape of those hemoglobin molecules. But for our purposes, it's just law of mass action, just probabilities. Yeah.
Does that explain why you have a 30% return, like return of oxygen? The oxygen concentration on returning blood, I think he says around 30%. Is that why, just as the concentration of oxygen in the blood as it's being diffused, more oxygen bound probability it's going to diffuse more? Is that why it just holds more and more and there's more hemoglobin to regain it?
Let me try to rephrase that.
Yeah.
So your question is, why is it that we only relinquish 30% of the oxygen bound to the hemoglobin as it runs through the circuit from the left side of the heart to the right side of the heart? Is that fair?
Yes.
Okay. The reason is that partly it's going to be, we'll see a little later, that there's this cooperative binding of oxygen to the hemoglobin. And so it's easier to lose the first one than to lose the second. Just as it's easier to load the fourth one than it is the third one. So it's a cooperative interaction. So that's part of it. But you can also get rid of more of that oxygen, and that's characterized by the blood flow through the coronary vasculature. Blood flow through the coronary vasculature will consume 85 to 87% of the oxygen. And those are the same hemoglobin molecules. They're the same erythrocytes. And so it's just that under those circumstances where those mitochondria in the heart are using all that oxygen, the local concentrations of oxygen are dropping. And so the law of mass action has them leave, and they drop, and the levels get so low that you can overcome that cooperative binding curve and basically strip all but 15% of the oxygens from those hemoglobins in the heart. So under the right circumstances, you can pull all of that oxygen, almost all the oxygen, off the hemoglobin. But through the systemic vasculature, you only use about 25 or 30% of it.
Okay.
Okay.
Thank you.
Anything else? Yeah.
If you mentioned this earlier when we were talking more in depth about anemia, I missed it. So if you had an iron deficiency, would that be sufficient to cause a buildup of like bilirubin and biliverdin?
Okay, let's separate these two.
Okay.
If you're deficient in iron, then your hematocrit, rather than being 42 or 45, may be down around 35. If you get much below 35, you're feeling pretty bad. And so you still have a lot of hemoglobin that has to be turned over every day, even when you're anemic. And so you still will generate the bile, and you still will need your liver to get rid of it. And if your liver isn't functioning, you still will get jaundiced, even if you're anemic. Now, if you have iron deficiencies, in the old days, they used to tell people to eat liver, because calves' liver, particularly, was storing a lot of iron. I mean, we store iron in our liver, so it was a rich source of iron. There are better supplements these days than eating liver, but yeah, you still get the iron. Did that answer it?
I think so.
Okay. Anything else? Okay, we'll see you again tomorrow then.